and then do the sequencing using the Sanger sequencing. Um, and then with the slight advanced method by shotgun sequencing done by Celera Genomics. Then they use a shotgun sequencing in which the sequencing is done only at the beginning of the sequence, and then the computer algorithm will match the overlapping and slightly the overlapping plus some other sequence. So that will be the techniques of doing the sequencing. So you imagine that process of doing the sequencing. And of course, in the next generation sequencing, instead of doing uh, one fragment going doing using the Sanger sequencing for 500 base pair length, and then you have to design new primer and so on. But with the next generation sequencing, you can do it much faster, and even at the moment we can do like millions of sequences in one reaction. Okay, so now you have all the genome sequencing. So we have seen different organism have different genome size. So and then we have discussed or came across, uh, from the complexity simple organism to the more complex organism, increasing genome size. But then you cannot always say that genome size is proportionate to the complexity, because we have this paradox, okay, C-value paradox. So I mentioned about what is the C-value paradox and so on. So now, and then after that, want to we want to learn more about gene in humans, for example, but we cannot do testing in humans. So we done sequencing in other organism, and analysis have shown, for example, similar sequence between the uh, gene in human and gene in some other organism. So we can now start doing research on certain human genes, but we do it in other organism. So, so that come into play this functional genomics. So functional genomics analyze all genes in genome, and then based on that, we want to determine the functions of the gene, and also how it is controlled, how they are expressed, for example. So so based on that, you can answer a lot of question in terms of about the gene expression, in terms of development. For example, you might want to ask when is this gene expressed? Is it during the very early age of the fetus, or at the very late in the age of an individual? So you can learn about that. Okay, and then based on that, what gene I express when organisms are developing. So those can be done using the sequence that we have, sequencing that we have done for any given organism. All right, and then current functional genomics rely on molecular biology, lab research, and computing in order to do the analysis, especially on the sequence part. All right, so functional genomics um, with the when the sequence have been obtained, so you just imagine you have one book containing all the sequence of ATGC arranged in certain way, okay, certain order of appearance. So based on that, you want to find out. So based on those sequence, where are genes, where are regions that do not have genes at all, and okay, and then you want to align the sequence with database to determine the matching. For example, you want to compare the current genes with genes that has been genomes that has been sequenced from other organism. All right, and then based on the sequence, you can predict the structure of, okay, you can, because based on the sequence, you can uh, write the computer algorithm, computer programming to convert it into proteins or amino acid sequences, and then uh, write a program to further describe how they are folded, become a functional protein. So you can describe function of the gene, for example. So that is in the functional genomic. And then describe interaction between genes and gene products in cell, between cells and between organisms. So this one, for example, you want to learn about certain genes that are expressed but not as a phenotype, but that gene product is being used to influence the other genes, for example. So you have interaction between genes and gene product in the cell. Okay, and then you can have interaction with products between the cell and also between organism. And you can also consider phylogenetic relationship between certain organism to the other. So based on, for example, you have specific amylase enzyme. So you take amylase from human, amylase from bacteria, amylase from yeast, and so on. So you can arrange in an order to say that, okay, the sequence from bacteria is considered as the ancestor, and sequence that found in certain plant and in certain organism are actually derived or mutated from the original sequence that was found in bacteria, for example. So that is how you can do in functional genomics. All right. So now you imagine you have done the sequencing example like I have shown in the previous week, whereby you have arrangement of a sequence of the nucleotide. Okay. When you have the sequence of the nucleotide, you do not know yet what is, where is the start codon, stop codon, and so on. So after you have identified where the start codon, where the stop codon, and then you identify all those operating frame, everything. So those process is called annotation. Okay, until you identify the gene, uh, the role of that particular gene, so on. So the entire process is called annotation. So annotation begin the process of assigning function to genes, especially protein coding genes, using a computer algorithm to search both strands for open reading frame. So if you still recall, last week, I shown you the example of the nucleotide ATGC and so on. So you have the 5' to 3', and then you have the 3', the TAC over here. So 3' to 5' the other way. And then based on this, you can take this, convert into amino acid, for example. So every 3, 1 amino acid. And then every 3, amino acid number 3, and so on. So you convert the sequence from the nucleotide into amino acid sequence. And then you can also write a computer algorithm in addition to just doing the process of translating in silico of the sequence of from 5' to 3' of the main strand or the leading strand. You can also do it on the lagging strand this way. So this one will also convert into amino acid sequence, for example. Okay, because this one moved from 5' to 3', the other way, right? So amino acid also should go the other way around. So based on this, then you will be able to determine the series of sequence. And of course, some of them after you every 3, and then eventually you might find one stop codon. Okay, for example. So you have stop codon, and then you go again and have another stop codon. So it refers back to what I mentioned last week. And then for bacteria, it is quite straightforward because you have to, you remember last week in terms of gene density. So every 1 kilobase, you find one gene, for example. So there is no region that do not show any gene. So for eukaryotic organism, it a bit complicated because the uh, the presence uh, of the sequence that do not code for any gene, for example. And then in human, even in certain in organism, there is also a possibility of certain sequence, for example, have intron. Okay, for example, in human, intron in which if you have a very long chromosome, you know that this region is a region that contains genes. So even within that, within the region that has gene, there is also what you call intron. So intron has to be removed first before you stitch up those that from. So eventually when you remove the intron, for example, the sequence actually is this one, okay, and then this one, this one, this one, this one, and this one, and then you have to add, you call that, you know the sequence, then you continue, the next sequence is over here. You ignore the region that is from here until here, okay? So that is the intron. So based on that, then you have to write additional line of programming, okay? Within the sequence that has been obtained over here, for example, this within this sequence, okay? Then from here until here, you when writing a program, you instruct the programmer, for example, you have also to find where are the introns over here, and then the programmer have to write a program, and then the programmer will ask, so how do you identify which part of the region that has gene? How do you identify it is an intron, because they also look the same, right? So how are you going to identify which part of the region is the intron? Then scientists have to go and work again. So how can we identify intron within the region of the genomic sequence that contain gene but has intron inside, and we don't want the intron. So the programmer asks back to the scientist. Scientists have to go back to the lab and find out. Oh, this programmer so ask so many questions. So we need to find out how to do it. Okay, and then you tell back to the programmer. Okay, you go and search in the literature review of people who has been doing work on introns. You find out what are the sequence of the intron. Then programmer also go and do programming, write the program. You know, just like you know, like in Jason Bond, that CIA analyst is writing program to hack certain thing and then find out. So programmer will then find out, oh, okay, based on the study that was done many times by other people, we have the sequence of intron usually start with GT, and then we have several sequence and then ends with AG. Another people doing the work on intron also find that the front of the intron will start with GT, ends with the AG. So out of thousands of different introns, it looks like all or 90% of them start with the GT and ends with the AG. And usually the intron have a certain size. Okay, so then the programmer will then write program, okay, find a sequence that start with GT, ends with the AG within, let's say, 20 nucleotides. So this will be the candidate for the intron. So that is how it is done. So once you identify that, then the program will run and find, oh, this one got GTAG, and then got GTAG, GTAG. So they know that this one is the intron. So if we want to take the sequence, we take only the region that do not contain this sequence. Okay. So, and then based on that, they will analyze. Okay, so the focus on sequences most likely to encode proteins. And then a minimum size of open reading frame arbitrarily set and shorter sequence are not analyzed. So again, as I mentioned, what was done in the what was described previously last week. Okay, and then based on that, now you have a genome sequence. You can narrow down into into sequence. You know, you have very long DNA chromosome, and then based on that, this region only contain the DNA, contain the gene sequence, and then you remove all the introns within, then it is only very short. Okay, after you remove, so it is a very short sequence. And then you want to compare this with a sequence that you obtain from other organism, from rabbit, for example. So compare this with this, okay, and then see how it looks. Okay, so that is one. Okay, another one, another way is that sometimes they compare not only with one, but you just feed this sequence into a computer that programming that was done, in which they will then go to the database and then do the searching and come up with the similarity of sequence with your result of the sequence. So, for example, is the blast search that you see over here. Basic local alignment search tool. So you will learn more in your bioinformatics later on, but basically what it is done is that you have, for example, 1000 nucleotide sequence of your from your genome sequencing. You want to find out what is the function. So instead of you doing the comparison with the rabbit sequence, like with your sequence you do if you do manually, you take this, you put into BLAST. BLAST will search not only with rabbit but with other organism sequences that has already that is already present in their database, and it come out with the result. Okay, so BLAST is the program, the basic program that is usually used by molecular biologists, the geneticists, when they are working with the sequence. All right, so that is how it is done. And then you can search the sequence, either DNA or amino acid sequences. Okay, so you have sequence in nucleotide. This one, right? You remember, you imagine you do a genome sequencing, you got the ATGC sequence, and then you do BLAST. So the BLAST will search with other nucleotide already in the database. Okay, but actually in the database, it contains not only the uh, BLAST, the nucleotide sequence, but there is also database that contain protein sequence. So you can search your nucleotide sequence after you done the genome sequencing and process to remove intron and so on. You got a clean gene sequence and you want to find out what is the function of the gene, that this gene. Then you feed into the BLAST search. So the BLAST search will go into database, compare with nucleotide that is present in the database from other organism. And in addition to compare your nucleotide sequence with the nucleotide sequence already present in the database, this sequence can also be compared with the amino acid sequence. Remember? So your sequence in the nucleotide form, arrangement of A, T, G, and C. You want to know the function? You copy paste into the BLAST search will search. And then it come out. And then when that BLAST programming is doing the search, where you cannot see, it is actually searching through database of nucleotide from other organism and also database of amino acid sequence. So this is what is meant by this, okay? But amino acid yield more specific information since there are 20 possible matches rather than just 4. forget about this a short while. Okay, 20 possible matches rather than just 4. But question to you: how do you search nucleotide sequence and find similarity with the amino acid sequence? How can you do that? Because for example, in the this one is in my let's say this one is a database, nucleotide database, ATGCCGAA like that. But this one nucleotide, protein database contains sequence of amino acid, methionine, adenine, phenylalanine, aspartic acid, what else? Glycine. So the letters are representing amino acid. So how are you going to take your sequence in form of the nucleotide, ATGC, and search in the protein database? Anyone can attempt to answer that question? Anyone would like to answer that question? Okay,
because we know how the nucleotide correspond to the protein, so we can match the nucleotide expression order to the protein.
Yes, very good. I got several answers here as well. Convert to amino acid first. So what happened is that where is this? The ATG sequence that you have over here is first converted into amino acid sequence. Amino acid 1, amino acid 2, and so on. Okay, so actually what happened in the computer, in the program, when you put this one into BLAST, BLAST will then convert your sequence into amino acid, and it then search with this one. So BLAST will actually take this sequence, one, it search compare directly with the sequence of nucleotide in the database, but at the same time, BLAST will first convert your sequence into amino acid, and then it will search based on the protein database. So it will provide results. When the results come out, it will provide either in the form of similarity using the comparison with the nucleotide database, or it will show similarity compared to the amino acid or protein database. And usually, when you use the sequence that you want to do the search in the database, the result that come out show that the similarity that is more specific is usually obtained with the protein database or the amino acid database compared to the nucleotide. The reason being, when you compare at the nucleotide level, if this one is, if your sequence is, for example, also, your sequence is also okay, the light blue is your sequence, so your sequence is A, T, C, 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 G, A, for example. So if you compare the database sequence, the dark blue compared to the your sequence of the light blue, you can see that there is a differences at this one. So the program will tell that, okay, this one is already one nucleotide difference. But when you convert this ATCCG, for example, this one is ATC, for example, convert to M, and then GCG convert to A, and then GA what? translated, not convert, ATC translated into M, and then this one translated into A. The other codon translated into F, for example. Okay, and then with this one is the database sequence, right? And then with the sequence, with your sequence, ATC also is translated into M. CCC, you see, this one is CGC at the in the database. This one your sequence CCC, different, isn't it, at the nucleotide level? But if you recall, we have how many? 64. What? In the genetic code, in the first year, 64. The number of 64 usually what is associated with, consisting of, let's say, if I do the equation, it's equals to 61 plus 3. Okay, now can you imagine something or not based on this number? Anyone? Anyone? Can you imagine something or not based on that number? Let's see in the chat. I want to.
Is it the
Codon?
64 codons?
Yes, everyone who managed to answer that, well done. I think the others who didn't answer also might now recall you have learned about this, right? So you see that 64, 61 codon plus 3 stop codon is the number of codons, isn't it? So number of codons representing how many amino acids? How many amino acids we have altogether? Anyone? Let's see the chat. Anyone answer?
200 plus.
Only one answer correct at the moment.
20, sir?
20, yes. Representing 20 amino acid. Remember, in human, we have only 20 amino acids that are arranged differently to make into different enzyme. Okay, you remember that. We have only 20 different amino acids that are arranged differently to produce millions of different proteins and enzymes. Okay, so you imagine we have 64 codon in terms of the nucleotide arrangement codon, but we have only 20 amino acids, meaning to say there are amino acids that are represented more than once by the codon, right? Wobble hypothesis, remember? For example, only 2 amino acid that is only represented by 2 different amino acid, and each of them is represented by a single codon. Okay, one is methionine, the other one is phenylalanine. This one is represented by ATG. Phenylalanine, I cannot remember, but these are the only 2 that has only a single single codon representing 1 amino acid. But the others like glycine, threonine, they are represented at least by 2 codon, right? So in this case, 2 codon representing 1 amino acid. So there is a possibility like these 2 codon representing 1 amino acid. So in this case, for example, like CCC is representing the letter A, alanine, okay? And then CGC is also representing A. So you can see, although at the nucleotide level, they are different, but when being translated, they represent the same amino acid, right? So it means that at the amino acid level, they will yield more specific because this one is also MAF. This one, although the sequence at the nucleotide level are different, at the amino acid level, it is also MAF. So you can see that the information at the amino acids yield more specific information. So that is why this statement. Okay. But sometimes no convincing match is found because limitation to current database. Okay, meaning to say that even until now there are certain sequence that we do not know what the function is for that particular sequence. Although we know that it is very highly likely to be genes because it has start codon, operating frame very long, and then stop codon. So it matches the characteristic of a proper gene, but we don't know the function. And people or scientists is still investigating that. Okay, so that is what is meant by limitation over here. I cannot find the eraser. Okay. So, any questions so far? Any question? Any question, anyone? So, based on your sequence, it provides clue to the new protein function and the evolution of its gene. Again, as I mentioned, once you have the sequence, then search database, you obtain the result. So from other organism, so you know the function, that is one. Secondly, you can take the result from many, because when you do the blast search, it will come with a sequence from many different organisms. So you can take those sequence and then you can do analyze and see the phylogenetic relationship. So you can say that, okay, this one is the evolution of this particular gene when you compare with the similar gene from other organism. Okay? Let's see if anyone got question at the moment. Tak ada. Alright. So, do you have you come across the blast search or not? Anyone? Have you come across the blast search?
No, doctor.
No. So do you have, can you imagine when I mentioned about this blast search and so on? Any
Yes,
comment?
doctor.
Well, you can imagine, right?
Yeah.
Okay.
It's
Never
like
mind.
using Google to search what
Yes,
certain
yes.
sequence codes
Correct.
for.
Good. So you can imagine that. That is very good. Okay. Okay. I'm going to share one website on the. I'm going to stop sharing for a short while. Okay. Okay, and then I'm going to share internet. Where is this sharing? Okay. Chrome. Okay. Can you see now? U.S. National Library of Medicine, NIH. Okay, so this one is the
Yes.
very old database. Even when I was doing my PhD, they have started with this, but of course the what you call that, the appearance are slightly different at that time, but they are still using this. It's called NCBI. So this one is a blast search. So you can see over here. BLAST refers to basic local alignment search tool. I'll go very fast. So many different BLAST. So you can see over here. When you obtain your genome sequencing result, okay, you are in the lab, you do a sequencing, you got your result, your nucleotide result. So you can compare with your nucleotide. So you can say that, okay, I just want to compare my own sequence with nucleotide only. I don't want BLAST to go pandai-pandai buat protein. So you search this one. But then you also would want to have your sequence translated by BLAST and BLAST search protein. So you do BLAST X. Okay, and then you can also do T BLAST N. Okay, for example, some people do only protein research. So they have protein amino acid sequence, but they don't have the gene sequence. So you can also, a program has been written in which using the sequence of the amino acid, they work backwards to predict the sequence of the nucleotide. So your nucleotide can also be searched using that, but usually we use the blast egg because of the reason that I mentioned just now. Okay, you can also do protein blast. So with the nucleotide blast, so if you click on there, similar like what you are using in Google, so you can copy and paste. For example, if you imagine your sequence in the Word, in the Microsoft Word file, so beginning one whole page of only letters ATGC, one whole page. So you copy and then you 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 paste it over here. Okay, so you can either copy and paste, or sometimes you can also upload the file. Okay, and then based on that, you can set some of this, and then you just do the blast, and then it will start to search in the database what is the sequence that is similar to the sequence that you key in just now in their database. So it keep on refreshing after, every second, every few seconds. Okay, it will keep on refreshing, and then it will generate results. So it will tell you what is the potential role or the function of the sequence that uh, you uploaded for the search just now. Okay, so that is how it is done. Okay, any question so far? Any? Okay, no question at the moment. Sure, tak ada question. Alright, so I'm going to share again. So the result will look something similar like this. Okay, but of course, because my sequence is very small, so even when I have typed the sequence, the random sequence, so that sequence similarity is shown to be similar to this wheat, Hodium vulgare, wheat, and then in Pseudomonas. So this one will be similarity of the sequence that I have typed just now with this. So graphic, you can visualize. So this one you can see 100% similarity with what is in the database. Okay, so but if you have thousands of sequence, then you can see that some of them do not form a line. So it will be different because of the different similarity. Okay, so and then based on that, you'll be able to tell how good is the similarity. How good is the similarity based on the value over here. Okay, for example, from this you will know how good is your the sequence similarity. Of course, that one you will learn later. Okay, so but that is uh, the search that you can do in blast search to produce the result, to give an idea of what your sequence from the genome that you have sequenced just now. All right, so that is sequence similarity. So based on that, you can search your entire sequence, and then the entire genome that you have sequenced, then you will be able to determine what are the roles of the genes that you have sequenced. Okay, so as the database grow, so does our knowledge on gene function. Okay, so we will be able to tell. Okay, for example, in human, we don't even need to do experiment of gene sequence that we have sequenced, but we uh, compare with the experiment of similar sequence in other organism. If the similarity of the sequence is very high, looks like there is no difference, then you can more or less say that the function of your gene is similar as the function of the from what has been described in that other organism. Okay, so we determine the function based on the sequence similarity. So based on that, you can say that, for example, here, current distribution of knowledge about genes of yeast. So this one, you imagine, genome sequence of the yeast hasn't been known before, and then you have now completed the genome sequencing, and then based on the genome sequence of the yeast that you have done the sequencing, you feed into the BLAST database, BLAST search, and then it comes back with the result. So it will tell you how many sequence show similarity to what gene. So out of that, it will show how many open reading frames and so on. So based on the open reading frame, here it says that 30% of the open reading frame of your sequence from the yeast have non-function, meaning to say that based on comparison with study that has been done in other organisms, they know that the gene in yeast, 30% have non-function. And then 70% remaining, so out of 70, they divided further. 30% encode protein that either has a homology to protein of non-function or has domain related to functional characterized domain. Okay, so this one is that non-function, sorry, yeah, I slightly mix up. Non-function means that work has been done in yeast, the gene in yeast, and they put in the database. So 30% is based on the study already done on yeast. Okay, so 30% non-functions of the yeast information, either protein or the nucleotide. Another 30% is the one that is similar to sequence that has been characterized in other organisms. So homology to protein of non-function, so which of which studies were done in other organism, or sometimes even if it is not entirely. Okay, for example, you have sequence that is okay. This one is the length of your sequence. Okay, and you feed into the BLAST search, and then search of the database results show that similarity of your sequence is only at this part, at this region, with the region from, for example, this one is your yeast, and then this one is from a rabbit, for example, but study has been done. So similarity with regions on this proteins on rabbit. So you can see similarity at this part only, this part only. So this one is what is meant by domains related to functionally characterized domain. So they know that this has been characterized, and within the characterized protein function, similarity with your protein sequence is at this region only. Okay, at this region and this region. So based on this, you can also deduce that your protein has a function that is similar to the rabbit's protein based on the function similarity. Okay, so that one can also be done. So that is how we determine the function of the sequence after we do the genome sequencing. Okay, and out of that, out of the 70%, 10 are function unknown. We do not know yet what the function is. They have homologs. They have result when you fit into the BLAST. They come out with showing similarity of sequence obtained in cats, for example. But then those who studied cat sequence, they also don't know what function of that gene in cat. Okay, so function unknown genes. Even they have homolog, means that similarity with other organism in the database, but in that homologs, they are also unknown. So groups of homolog genes of unknown function you call orphan families. Okay, meaning to say that they do not belong to any protein function group, so they belongs to on their own. They are considered as yatim piatu lah, because they don't have similarity with other known functions. So they are the unknowns grouping. Okay, and then 30% of the operating frames have no homologs in the database, meaning to say that they don't have similarity with, of course, they they don't have similarity with what has been described from that specific yeast, and those in the database also no similarity, so they have no homologs in the database. And out of this, some of them 6 to 7% may not 6 to 7% may not actually encode for protein, meaning to say that it looks like a protein, but we do not know, and most likely there is a possibility that they become like ancestors protein, where ancestor gene whereby they don't have function. Okay, they do not function in the organism. So this one, sometimes they are called single orphan. Okay, so every genome sequence contains function unknown, even in human. Okay, but as database are expanded, meaning to say that as people are doing research all the time, so every time results are obtained, they put in the database. So database are expanded. So the number of unknown should be decreasing. All right. So this one is an example that I take from the NCBI a couple of years ago. Okay, assembly uh, in 2011. So usually for Saccharomyces, usually more or less they are almost exhausted in terms of doing the sequencing, because it is already been completed. So based on that, so you have 12 million base pair of the Saccharomyces. So that is the genome size. Okay, and then they have updated. This is the database version. Okay, uh, and then they have updated many times, and then you can see coding genes out of this total sequence. The coding genes are 6,600, so almost 6,700. So you, and then you have a non coding genes, and then they are all small non-coding genes, meaning to say that the lengths are quite small. And then you can see another category, it is pseudogenes. So this one is another grouping where it is similar, like this function unknown, but sometimes they are considered pseudogenes because of it. There is another gene that looks similar to it, and then they do not function. Okay, so that one is a pseudogene. Okay, and this one I think. Okay, so if you do like this one, Peter Russell references. So this one uh, is describing here. Okay, so homologs to experimentally characterized function. So they have studied, okay, and then recognizable motif or weak homologies, which this one is describing this, okay. Domains related to this one, domains related to functionally characterized, meaning to say their similarity is only at certain region. So that is why weak homologies, because their homologs, their homologies is are only at certain area, okay. And then orphans, and then single orphan, and then questionable goes into the pseudogenes, okay. Possibility of this one is not actually a gene function. So sometimes, for example, it does not have start codon. It does not have a transit peptide usually found in front of the gene, but then the entire open reading frame is very long. It has stop codon, for example. So it looks like a gene because of a long open reading frame, but then they don't have a start codon, don't have transpeptide, which is used to transfer to another organelle within the cell, for example. So that is questionable ORF and also the pseudogenes. All right. So based on this, you can see some of them has function and some others do not have functions. we cannot assign, isn't it? So when you do a genome sequencing, you want to find out what your sequence are, so you do all the processing to find out, or based on this, you determine there are 6,000 genes. And of 6,000 genes, we ask the computer programming to do the search with the database. They come back, and out of those results, you can see that some of them do not have similarity, but in database also, they don't know what the function is. So you want to find out further what is the function of that gene. So then you can start to do experiment. So you want to assign gene function to that unknown. So meaning to say you want to start doing the experiment. So you want to assign the gene function based on the experiment. So how to do it? One of the simplest method to do it is that because for example in the yeast, when we do the sequencing, coding genes 6,700, and out of that there is one that is function unknown, but you are interested to find out. So what you can do, the simplest one is that you kill off that specific gene in the Saccharomyces and see what happened to the Saccharomyces. Okay, for example, if you, let's say you um, identify this gene that you want to find out, so you want to kill that gene so that it is not functioning in the Saccharomyces. So when you want to kill, we don't say kill when we are working with the gene, we call it knockout, or we delete the gene, and then when we delete the gene, that gene is not function in the Saccharomyces, then we say that that gene is knockout, or sometimes we call it KO. Okay, knockout. So usually we call it knockout. All right. And then how do you want to delete the gene in that E. coli? Because now I'm using E. coli as an example, but you can always imagine other organism. You can always imagine I want to knock out gene in sago plant. I want to knock out gene in rice, for example, like what we did in the lab, for example. So how you can do knockout? There are many ways. Probably you have come across CRISPR, CRISP, CRISPR Cas9. Okay, not Has9, not Has9, but Cas9. Okay, Cas9. It is a protein. Okay, it is a protein that can do a specific knocking out of a gene. Okay, so this one is more or less the latest way of doing a knockout of genes, and then the what do you call that, inventor, I think the inventor of this won the Nobel Prize for the work that they did, because but of course they didn't invent, like you go to the lab, you said, oh, today I want to come up with different method to knock out the gene lah. But actually this one also come from the genome study. They find out certain protein, and then this protein they work back the sequence. So they take out the sequence, they produce it in E. coli, and then they investigate what is the role. So it can, if you, for example, this protein is something like that, and then if you can add sequence that you want, for example, this one is your sequence on in the sago plant or in the Saccharomyces cerevisiae. It contains ACCGAG, for example. So what happened is that using this information of sequence of this unknown gene in Saccharomyces, I want to knock out this gene and see what happened to the gene. So what I can do is that I can insert this sequence ACCGAG over here in this protein. So this protein will then go into the Saccharomyces, okay, and then it will go over here. Okay, and then it will go, and then it will find the similar sequence, and it will attach to it, and then it block. For example, it block this gene from functioning. So now in Saccharomyces, you knock out this gene. So when you knock out this gene, what happened? You find out that this Saccharomyces now, instead of being rounded, now it becomes jagged. The membrane, for example. So you know that this gene, actually the function is to provide a smooth surface of the Saccharomyces. So by knocking out, you are able to determine the function of this gene. All right. So this is what is meant, how it was done. Okay, but of course, the more simpler method is by using PCR to do gene knockout. Okay, so how to do that is quite straightforward. So this one is, for example, the gene that you want to knock out in the Saccharomyces. So it is a very long gene in Saccharomyces. So you want to knock out this one. Okay, you want to knock out this one, but then the way to do it is that you replace this part with the part that is not having the same sequence. So this one, the sequence will not function anymore. So how you replace it? By homologous recombination concept. You remember in mitosis, meiosis, chromosome align, isn't it? So when chromosome align, they are based on the usually when chromosome align, this one is the diploid organism. Usually the sequence are very similar because they are the same chromosome that exists in a pair. And then if you recall the meiosis, even for example, after the meiosis 1, where the chromosome align, and then the crossing over took place, isn't it? Then you have recombinant. So the same concept of what you learn in the recombination at the chromosome level, we can do it at the DNA level, because actually it's the same process. So you do a synthetic DNA, you ask the machine to produce the DNA that contain sequence at this region similar to the yeast, at this region similar to the yeast, but those in the middle are different. Okay, and then you take this fragment. Okay, you take this fragment. You this one is the yeast. Okay, this one is in the yeast. And then you do a transformation to put in the yeast, and then within the yeast, the chromosome tends to align. So it will align and then do the recombination. So this one will go into the yeast genome, genomic DNA. And then when the yeast undergo the process, then it will start transcription, but then the original gene has been removed before, has been removed. So now the gene is not functioning. So you have a knockout mutant. All right. So that is how you do it. So this one is the what you call that, description of the work being described over here. Okay, so you can see over here, they express kanamycin resistant instead of gene under study. So what happened is that, you know, when you swap places, you want to find out whether your modified gene has gone into yeast or not, isn't it? So you want to find out. So how are you going to determine? Because yeast cannot tell you, sir or doctor, I don't, now my original gene, this one, has been removed. Now I have foreign gene inside. They cannot tell you, isn't it? So one way to tell is that by inserting the kanamycin resistant to take over this region, when the yeast now has the kanamycin resistant gene, which replaced the original functional gene. Now, the yeast has new function, which is having resistant to kanamycin. So you take this yeast, you grow in kanamycin, and it can grow in kanamycin because this one produce anti-kanamycin product, for example. So you know that if the yeast is now growing in kanamycin, it means that the genome of that yeast has been replaced at that particular region with the kanamycin, so that all yeast that is growing in the kanamycin plate, so this one yeast growing in the kanamycin plate, are actually yeast that contain the knockout. We call this a knockout mutant. Okay, knockout mutant gene. So you have a null mutation or loss of function mutant. So this is also called knockout mutant. You call a null mutation, or you can call loss of function mutation as well. Okay, and then how? That is one way to determine whether you have a mutant or not. The other way, of course, you can always do a PCR. You just do a primer over here, and then the primer will amplify this region. You take this and then you do a sequencing. Then you can confirm your sequence as well. Okay, so based on that, you systematically determine, meaning to say here, yeast knockout project, meaning to say by using the knockout method, you reduce uh, the number of genes that was previously unknown. So you systematically, so based on the using the yeast knockout technology, you delete each gene at once. So if you have, like for example, 800 genes that is function unknown, so you today you work with gene number 1 of FUN, and then next week you work with gene number 2, and so on. So you work one step at a time, systematically. Okay. So, but some of the genes, when you do knockout, and then you plate in kanamycin plate, the yeast couldn't grow. And then you might wonder, what about if I grow in the normal media? It also couldn't grow. So when that happened, it means that that specific gene, although we don't know the function, it is a very essential gene which gives a lethal prototype when we do modification. Okay, so based on that, those who can survive in antibiotic kanamycin, for example, actually 4,200 out of 6,200, so almost 2,000 that is unable to be grown when we do a knockout mutant. Okay, and then out of those viable knockout, this one 4,200, half of them show a detectable phenotype. So out of, okay, for example, uh, 6,200 are FUN, can function unknown, and 6,200, 4,200 are able to be used. Okay, 4,200 is non-essential, so they are viable, meaning to say that they can be uh, they can produce mutant, and out of this 2,100, okay, show a detectable, meaning to say out of this 4,200, when you do a mutant, another 2,100, you see there is no changes in their physical characteristic or the phenotype. Okay, so you do not, based on that, this 2,100, although they can grow as when you knock out them, but you don't know what is their function. Okay, so null alleles using this knockout mutant are usually used to investigate gene function. Okay, and then in human, we also have some function unknown genes. So what we can do is that we cannot knock out in human, see what happened to that particular baby, for example. It is unethical, isn't it? And then we know in mice, there is a similar gene. Okay, so we knock out the that gene in mouse and see what happened. The the eye become red color or whatever. So we know that that is the function of that specific gene. Okay, so that is how it is done. So genomic sequencing makes it possible to determine all genes that are expressed in a cell by analyzing total RNA transcript or the transcriptome. Oh, it comes transcriptome already. Okay, the transcriptome is an indicator of cell phenotype and function. And similarly, the complete set of protein is called a proteome. So you see the omics. Now it start with the omics already. Okay, transcriptomic, proteomics. Okay, so when you do sequencing of the entire genome, you call it the genomic sequence, the genome sequence. But if you isolate and uh, do the sequencing of the transcript, then you call it transcriptome. So total RNA transcript of the cell, transcriptome. And then sometimes you do a protein analysis, you call it a proteome. Okay, hang on, yeah. I want to see if there's any question. Good. What if the knockout gene already answer? Oh, okay, no, it doesn't give phenotypic effect. How do we know that it codes for them? I already answered the question, but actually still unknown, isn't it? So how do we know further? So what another way of knowing further is that you find that gene, you find similar sequence in other organism, and then you do in other organism. So you will be able to at least know the function of that gene as well. Okay, so that is why we have different model organisms where we can study same gene in different organisms. So meaning to say, if it is not showing any changes in one model organism, we can work in another model organism working on that same gene as well. Okay, so we can use that. Okay, so now you have sequence of the genome, and then you feed into BLAST. BLAST will come up with the sequence information, and then tell you what are the functions. So you are annotating the sequence. Okay, but sometimes you do not want to work with the genome, because let's say I don't want to work with the genome, because if I have the sequence of the genome, then I have to ask someone to run a program to find out how many introns are there, and then stitch back those region, the exon, and to join back after removing the introns. So those exons now become a functional gene, and after that I do the further study on that. But I don't want to do that, because I say that I have some doubt with the work of that programmer, and then I am not confident with his programming capability. But I want to be certain that without the intron, the sequence that I have is the actual sequence in the cell. So in order to do that, I decided that I don't want to work with the genome. I want to work with the sequence that is only representing the gene. Meaning to say that how are you how are you going to work only on the sequence that representing a functional gene? So one of the way to do that is by studying the RNA. Okay, you recall the process of transcription, translation, and then you recall what is the difference between transcription in eukaryotic organism compared to the transcription in the prokaryotic organism. So you compare where you, if you recall, we have the process of gene being transcribed into pre-mRNA, and then that pre-mRNA is processed using spliceosome to remove the intron and stitch back everything together, done in the cell, and then it is exported out of the nucleus into the surrounding cell in order the mRNA to be translated, isn't it? So now we are actually asking the cell to do the work of, I'm not asking the programmer if I work with the genome sequence, but now I'm asking the organism itself, the cell of that organism, to do the work of removing all the intron sequence. So the cell will remove the intron and then join back all the mRNA to produce the mature mRNA. And then if you recall, that mature mRNA in the cell is the one that is going to be used for translation to produce functional protein, right? So you want to study, okay, now I want to study the mRNA. So that mRNA or the RNA, if you want to consider you study everything, then you what you call the transcriptomic study. Okay, yeah, intron splicing is happening in the cell whereby the introns are removed, right? So thank you. Yeah, you are correct on that. Okay, so that is how you do. So now there is another way, another group of scientists who wants to do that. They only want to work on the mRNA. And then how, then one of the other advantage of the mRNA is that, for example, if I have E. coli that is growing in this the room temperature, okay, and then but when I take this E. coli, I put in the fridge, the E. coli uh, uh, die. Okay, it becomes dead, not growing anymore. It is dead. You see, no function, nothing. It is dead basically. But then somehow I take my E. coli that is growing at the room temperature, and then I go outside playing badminton under the sun, just like people going to the beach, sun tanning, you know. While I'm playing badminton under the sun, the E. coli is exposed there. After several times taking in and out, it is my pet E. coli, for example. And then someday I decided that I want to put this in the fridge, and somehow my E. coli grow, you know. Now, why is it growing? So, you know, the UV light creating, making my pet E. coli become mutant, right? So that E. coli now can grow in very cold. So I have same E. coli that originally growing in the room temperature but die in the cold, and then the same E. coli but now probably have some modification. For example, growing in can grow in the in the fridge. So I have two sets. Okay, so what I can do is that I can compare and see what is the mRNA differences between them. So you can study the function in which you find that some mRNA are not expressed, for example. Okay, so you can study that by using transcriptomic, because you remember our mRNA, for example, our cell that exists that make into our head, into our finger, our hand, our lungs, our heart, our muscle, our intestine, for example, they have the same genome sequence, isn't it? Okay, but then at the transcript level, some of the gene are expressed in the lung, but that gene is not expressed in the muscle, for example. But and then I want to know which gene is expressed in the lung, which gene is expressed in the muscle. So if I do the genomic sequence study, there is no difference, right? Because everywhere we come from the same, we have the same cell. But then when you study at the transcriptomic level in the lung, different genes are being transcribed and translated. In the muscle, different genes are being transcribed and translated. So you can compare and see the differences. So you know that, oh, these are the transcript that is only found in the lung tissue. Then you know, okay, then you can take and then do further study on that. So that is what people are doing, including what we are doing in the lab. Okay, so transcriptome is a set of RNA molecule, including mRNA, rRNA, tRNA, and other non-coding RNA produced in one or a population of cells. Okay, just like I mentioned in lungs, in muscle, in whatever tissue you can imagine. So transcriptome changes as the cell responds to stimulus, just like I mentioned just now, my pet E. coli, for example, and move through as the cell move through its cell cycle. So even through the normal cell cycle, even when they are not growing at different condition, at different life cycle of the cell, different RNA are being transcribed and translated. Okay, so you can study that as well. Okay, so one of the way to study that is by probe array to study gene expression. So you can see over here, gene expression are studied at the DNA or RNA level. Okay, in this example, for this slide, using yeast sporulation, for example. Okay, so here is the life cycle of the yeast sporulation. I'm not going into detail, but throughout the life cycle, you can take at two different stages, for example, and then you can compare in terms of what RNA is being expressed at different time. All right. So you can have a reference sample and you can have experimental sample. Okay. For example, you can take uh, one is at the early stage sporulation. And then you can take as the, for example, this one as the experimental. You want to find out at the early stage, what is the gene being expressed compared to the at the spore maturation, you call it a reference sample. So you can have a reference sample of the from the happen of the mature mRNA, and then you can have the mRNA at the early sample. So reference and also early sample, as which is the experimental. Actually, you can decide which one, because here we want to compare one condition to another condition. So what we can do is that we can take the RNA from this time and extract and remove all the genomic DNA. So your collection is only the mRNA. So you can see over here, yeah. One thing that I would like to also mention, transcriptomic usually refers to the entire collection of the RNA in the cell. Okay, as the mentioned over here, mRNA, rRNA, tRNA, and other non-coding RNA. That is the actual description of the transcriptomic study. But then, out of this, sometimes we only want, because we can do genome study and determine what are the genes. Okay, but then we want to know for specific genes or for all genes that we have studied through the genomic sequence, we want to know which gene are expressed during the early stage, which gene are expressed at the late stage. So we want to compare genes that are expressed and doing some function. So that is why, although these are transcriptomic study, we are now only looking at the mRNA. Okay, so we can take the cell at the early stage, break it up, do the mRNA purification. So you have mRNA early, mRNA from the late stage. So and then what you and then you recall, if I think I mentioned mRNA is very sensitive, because if you mishandle it doesn't mean that you slap or you throw something like that, but then you talk so much, when the tube containing your mRNA chit-chatting, mRNA tube is open, gossiping, and then your saliva goes into your tube, and then it can degrade the mRNA easily. So mRNA is very sensitive. Your hand also, somehow you hold at the tip of the tube and then you close, you shake again, then your polymerase on your finger or some other enzymes on your finger can degrade your mRNA. So mRNA is very sensitive. So we have to do something. We cannot work with mRNA because many stages in the experiment, if we use mRNA, at the end of the experiment, you might not have the mRNA anymore because all of them were degraded. Even at the room temperature, sometimes it can degrade without any enzyme. So hang on, yeah. Does transcriptome also apply in HOX? I try to recall gene expression domain. Probably you need to mention what is that. Can you elaborate what you mentioned? But basically, I think what you want to know is that you want to find out whether the gene is transcriptome can be used to study gene expression. Is it? So yeah, so this is what I am describing at the moment, how you can use mRNA to study gene expression. So okay, we continue. Continue first, yeah. So now you have early stage, late stage. You determine one of them as the reference sample, was one of them as an experimental sample. So what you do is that you need to differentiate between two populations or two groups of mRNA that you have. So what you can do is you can give a fluorescent dye to your mRNA. So each population of mRNA, the reference sample, the experimental sample, you can give a dye, a fluorescent dye, Psi3, Psi5. So you can have different color so that when you put under the detector, they will be able to determine. Okay, even when you mix together, they give certain color, so you will be able. For example, if you add them at equal amount, it will give certain wavelength. The the detector will detect, oh, equal amount. If you have this 30%, this one 70%, it gives different intensity. The detector can detect, oh, this one 30-70, and then another measurement 20-80, and so on. Okay, so even when you mix them, the computer detector interpreted by the computer will be able to determine the level of expression compared to one another. So when that happens, you can mix them together. Okay, mix them together, and then after you mix them together, you can hybridize on the glass slide. So each mRNA will go to specific position on the slide, and then depending on the number of copy for each mRNA, then it will bind to the same position. Okay, for example, if okay, and another thing similar like with the chromosome number when you want to do experiment, right? So when we extract the mRNA from one gene, meaning to say it originated from, for example, like a gene, this one, gene number 1 is over here, gene number 2 is over here, gene number 3 is over here. So when these are translated, transcribed into mRNA, you can have many transcripts originating from gene number 1. Transcript originating from gene number 2, number 3, and so on, right? So each tube here contains, if there are 10 genes in this hypothetical chromosome, all 10 genes will be transcribed, but each of those 10 genes will have different copy number depending on the requirement, the gene control, the gene expression level, right? So that is why. And then, so like for this transcript, it goes to position number 1 on the chip. Gene number 2 go to the second position on the glass chip. Gene number 3 goes to the third position on the last chip. And then, so if this one is labeled as a green color, for number 1, it will come here. Okay. Number 2, label with green color because this one is a reference sample, for example, green color. It will come according to the gene position, not according to the color. Okay, and then on this side, you have the red color, isn't it? With the red color, you also have the same organism, gene 1, gene 2, gene 3. So for gene 1, it will be transcribed as transcript number 1, transcript number 2, transcript number 3. Okay, and then it has the red color over here. So, and then when you mix together, they still exist as this one red, this one is green. So when you mix, so for number 1, it will still come to this position number 1 as well. Number 2 will come over here. Number 3, this one will come over here. This number 3 will come over here. So on this position, there will be a mix of green and blue. Oh my God, there's no blue over here. Green and red over here, a mixture. So depending on the amount or the number of the copy number of the transcript, then you can see a mixture of red and green on this position for gene number 3. And then this one mix of red and green for gene number 2, red and green for gene number 1. So after that, if you then bring this chip for detection by the laser, it can measure the intensity of the mixture of red and green. It will give different color. So it will tell, okay, for example, for gene number 1, it is highly, for gene number 3, it is highly expressed on the reference sample, which is the late stage, this one, where compared to gene number 3 on the early stage. So you can see the level of expression for gene number 3 is less compared to gene number 3 is less for the early stage compared to the late stage. So at the late stage, gene number 3 is expressed more. So they can be determined using the chip like this. So you just imagine if you have 1,000 chips, 1,000 gene, so 1,000 dots of the chip over here that can be used to determine the level of expression. Okay, one question. How can gene number 1 that is over here come to this one, gene number 2 come to this one, gene number 3 come to this? So same goes for this. 1 come here, 2 come here, 3 come here. Why 1 didn't come over here? So how do you determine, how do you set so that it becomes like that? They go to their own specific place. So for example, if you have 1,200 genes, each will go into its own specific position. So that is one question. How will you be able to ask the gene to go to its specific place? Anyone would like?
Separating by weight in the spinny thing.
Okay, that is one. Separation lah. Okay, another, some other answer, please. You have to imagine a lot, because as I always mention to the molecular biologists, okay, wavelength, number 2, wavelength, polarity. Okay. What else? Spinning, okay, separation, that one is separation. Okay, spinning, centrifugation. So that one belongs to number 1. So 3 options at the moment. How do you ask? Hey, number 1, you go to that position lah. Okay, what else? Takkan 3 saja. The gene knows. Okay, number 4, the gene knows. Okay. Come on, anyone? Gene knows. Okay, what else? But then I can ask you again. How do you tell the gene to know where to go? How does the gene know? Density, separation group. Okay, someone answered density. We go into separation, similar to concept to that. Okay, length, separation as well. Okay, short length, long length. We separate them accordingly. So separation. What else? So you have to imagine. I always told everyone who work with molecular biology area, you have to imagine a lot. So that's why when I teach, I would want you to imagine with a marker. Okay, number 5, marker. Oh, this one you can relate to the genome, right? Good. Length, okay, you based on separation. So, okay, 5. Give up already? No more answer? No more? Give up? Length, also separation. No, I addressed that already. Okay, no more. All right. Mapping is the marker just now. Okay, marker. All right, so good. At least those, hopefully everyone here can at least imagine what we have learned in the past few weeks, more or less. All right. So marker, you can relate to known region of that particular, on the chromosome, there's a marker, right? So you can use part of that as a marker. So mapping and a marker, yes, can group into marker for the mapping. So how you can ask the mRNA to go to its respective place, you have to remember that the DNA, okay, before that, here from mRNA, you convert the mRNA to cDNA by reverse transcription. So this one is what has to be done because of the mRNA is sensitive, right? So you convert the mRNA into cDNA from single-stranded to become double-stranded. So what happened is that based on your genome study, there are, let's say, you are interested to know that there are 120 unknown genes from this yeast sporulation, the yeast genome, 120 unknown. So, in every one, you know the sequence, right? So, because you know the sequence, then on each position, you 1 until 10, 11 until 20, and so on. So, you can put your fragment of the fragment of that gene that you want to investigate. So you have from the genome, you have 120 gene, for example. So based on that 120 gene, each of them, you have specific size or specific region on that gene that is not cross contaminating or that cannot be cross referred to the other 120. So it exists as single individual specific gene. So you put them on the position. So you have a specific gene on the position like that. So and what happened after that? Once you put your specific gene on the position, so they become a probe. So that is why it's called open reading frame probe generated by PCR. So you remember, you sequence the yeast, 120 is unknown, and each of those 120, you know the sequence, right? And that sequence that you know, you take specific region, and that specific region can be a marker, can also be a map. But that specific region, you make sure that do they do not exist on the other 119, right? Because if they exist at some other place, there is a possibility of cross contaminating. So what happened? You can do the amplification of that specific gene, that specific region, and they become probe, which you can do by PCR. And then you, for example, like this gene, okay, on chromosome, gene 1 over here. And out of this gene 1, this region, you can do PCR. Gene number 2, this region, you do PCR that you know. In other genes, this area do not exist. So you PCR this part, then you PCR this part. So when you amplify a fragment, this one, you come and attach here. This one, this region, you come and attach here. Of course, when you do PCR, is it single copy? Many copies, right? After 30 round, 1 million, isn't it? So you take a little bit of that PCR reaction, you put over here, and then it will stick to the glass. So it will just be like, you know, like what you when you see uh, in National Geographic seaweed growing on the seafloor. So it will grow over here. So it will grow over here, which is actually the PCR of your amplification of different. So 120 genes, they are at different position as a seaweed. And then, because when you do PCR, they are double-stranded. So you can treat them so they become single-stranded. How do you treat them so this probe becomes single-stranded? How do you treat this PCR probe to become single-stranded? Very straightforward answer. Come on. 10 seconds. 1, 2, 3, 4, 5. Hey, dah belajar dah ni. 6, 7. No one answer. Helicase. One answer. Good, but not the answer. But good try.
Transcript test.
No, not yet. Not transcript test. How do you make gene single-stranded? What make the DNA become double-stranded?
Unwinding, yes.
No. I got one answer. 2 answer correct. How do you make the DNA become double-stranded? With the existence of hydrogen bonding, isn't it? So if you remove the hydrogen bonding, DNA is still intact, the strand, but then they become single-stranded, right? So how do you remove the hydrogen bonding? Yeah, I got several people answering correctly. You just hit the DNA on that is now attached to your glass slide, which now they are called probes. Okay, so you hit that. You hit DNA to. You remember PCR? There is a at 94 degrees, it becomes single-stranded, so that the new primer can come in, isn't it? So same concept. When you heat up, then even though they are stuck to the glass slide, but then they are still they are double-stranded. When you hit them, this one still maintain. They cannot because they are totally stuck. But then each strand, when originally they are double-stranded, when you hit them, they become single-stranded. So when you become single-stranded, then later when you have this cDNA that contain the color, you can also treat them to make it into single-stranded. Then they will go into, they cannot directly float to come here. You know, just like the thing that you put in the beaker, you put liquid inside it, and then you put some thread, benang, then you just mix, so they just move around. When they find they can complementary base pair with this, then the probe, the red color here, and then this one, the green color. So equal give different level of intensity. But then in some other cases, more green. So more green. Then then it give different intensity. So that is how the sample come and bind to the probes. Okay. So this is what you call microarray. All right. So based on that, you can study and determine some genes are present during sporulation, some genes are present during late sporulation, for example. So based on that, you'll be able to say that, okay, gene number 1 is actually active during the early sporulation. And then gene number 20 are more transcribed more. So they are expressed, meaning to say that the protein, more RNA, more protein expressed. So they are more for in the late stage, for example. So you will be able to tell that. So based on that kind of microarray, show that 1,000 genes with altered expression during sporulation, but 50-50 repressed, meaning to say they are very low at level of expression, and another half is not expressed. Okay, so this is how you determine the level. So you determine the level of expression using the mRNA. Okay, so examples of study that currently use this technology. Many examples you can do that. Human in Drosophila during the process of changing. Okay, the characteristic. Okay, and then in cancer, okay, can also be used. Okay, transcriptional fingerprint that reveal distinction between certain types of cancer. So certain cancer have certain gene expressed very high. The other type of cancer, certain gene expressed very high. Okay, and then screening for genetic disease. Resulting from one or of many alleles. Okay, a patient's blood for 100 possible mutation of these BRCA genes that is associated. So you know that these genes are present in normal individual in at very low level in cancerous or potential to have cancer. They are expressed at other level. Okay, so many can be transcription can be used to transcriptomic can be used to study gene expression, but and then pharmacogenomics relates to you do the expression study but relate to genes that cause disease only, for example. So you call it pharmacogenomics. And then proteome is also you apply the same concept in which you have different cell or different tissue of this from the same individual, same species. They are expressed differently at different tissue. So you can say that, okay, this protein is expressed more during sporulation. This protein is expressed more during dormancy, for example. So this is the proteomic study that can be used uh, to determine the function of the gene. All right, so now it's 3:55. I think it's very long already. So I'm going to stop over here. So before we go off, any question that you would like to have related to either this lecture or if it is related to your assignment or anything?
Doctor,
Yes.
may I know when will we have our mid-sem exam for genetic?
Oh, okay. Um, Do you think you would want to have it next week? Is it possible next week?
Yes.
Yes
Can,
or no?
can, Doctor,
Can, Doctor.
Can, huh?
can.
Oh, can. Or another option, because my thinking is I would like to do it in week 8, actually.
Also can,
Can,
Doctor.
can
So
also.
can, yeah?
Can
Also
also.
can, also can, also can.
All right. So I think we will do in week 8. I will confirm that. And then hopefully we can do at, hang on, yeah. Let me see the number of participants today. 124. You all have 131. I know that some of you are not able to come because of other class. So there is a possibility if to allow for everyone to attend and not disturbing the other course that they would prefer to go at this time. I would like to do during the evening, let's say around 8 to 9:30, for example. Is it okay? Uh, no, that is the scenario. I think what I can do is that I will do a survey again, right? So that everyone can give response to that. So I do think that it's not fair for me to allow those who have class at this time. For lecture is fine because they can always refer to the video, but then for exam they have to be for the exam. So I think it's only fair if we do the times that I hope everyone would be present, which is during the evening from 8 to 9:30. Okay, so that is what I plan to do. I will do a survey again. Hopefully you can answer that. Okay, with regard to the question, we'll do MCQ. Okay, we'll do MCQ only. No, no essay or short answer. Only the MCQ. Hang on, I want to just pause the recording. So MCQ only. So anything else? Okay, the quiz. Someone keep on forgetting to look into the ellipse and always miss the deadline. So I extended the deadline again for those who didn't attempt the quiz. It's only 5 questions for the quiz, isn't it? Not that many. So please remind your friends